I'm going to be lecturing on the sliding filament theory today. Before we get into what the muscle's doing, I want to go over the synapse connection. Now, this was something you drew in Lab 5, so you should have some knowledge of it. I'm recording today because for my Tuesday class and also for you guys. That's what that is. All right, so here we go. So a motor neuron that innervates skeletal muscle looks kind of like this. Kind of funky. All right. It's got all these extensions. We're going to get into this in the next unit, but this is a motor neuron. Motor neurons carry motor information away from the central nervous system, away from the brain and spinal cord, and that information travels out to something that's going to respond. In this case, a skeletal muscle. Now, what's interesting is that every single skeletal muscle, which we know a skeletal muscle cell, every single cell, it looks kind of like this, right? It's a cylindrical cell with striations. Every single individual muscle, this is nuts, has a neuronal contact point, its own neuronal contact point. It's not like you innervate, one area and it just spreads through the muscle. It's not like that. Every single cell has a connection to the end of a motor neuron. Now, we call sort of the group of muscle cells that are innervated by a single neuron, we call that a motor unit. Sometimes you will have what's called a small motor unit. So maybe you have a neuron, and I'm just going to draw it like this, where the end of it branches into, say, three branches at the end. And therefore, in this scenario, you have one neuron contacting 3 individual muscle cells. That would be considered a small motor unit if you only had a very small number. 5, 10, even 20, that's small. That's a small motor unit. But sometimes this neuron will have 1,000 branches, 2,000 branches at its end, and contact thousands of individual muscle cells. That would be a large motor unit. And then, of course, there's everything in between. The smaller the motor unit, just so you know, if you want to get sort of an idea functionally, the smaller the motor unit, the more precise and delicate the movements are at that muscle. So like your eyes, for instance, your eye muscles can move your eyeball fractions of a distance, right? You can focus on 2 dots that are very close together because your eyes can go back and forth with these fine little motions. What you do with your fingers, like being able to pick up tiny little pieces of lint or the dandruff out of your friend's hair, right? That's a fine motor unit that you're doing with your fingers. So that's going to be small, a small motor unit. And then your really big muscles, like your thigh muscles and your butt muscles that allow you to do big things like walk, those muscles, like your quads, your hamstrings, your glutes, they're innervated by large motor units where you have thousands of cells being contacted by one motor neuron, okay? And they tend to be more sort of big, gross movements that don't have a lot of coordination compared to like, okay. So that's the first thing I wanted to just explain is what a motor unit was. All right, so what we're gonna do now is we're gonna zoom in on this connection. The connection between the end of the neuron, that's called the axon, this is called the axon and the axon bulb, and the point where it contacts the muscle cell. Okay, so that's what I'm gonna draw now and kind of zoom in on so we can understand what happens. I'm going to draw it bigger. So we're going to say this is our axon, the cell body be way back here. And then we're going to draw just sort of showing where it's sort of contacts the cell. I'm just gonna kind of sort of push it and see where it's contacting its muscle cell. And when we're talking about skeletal muscle innervated by a motor neuron, we call this a neuromuscular junction. And that's another way of saying a synapse. A synapse, by definition, is just the functional connection between two communicating cells, okay? Which tend to be a neuron and something else, okay? Synapse between motor neuron and skeletal muscle cell. Okay, that's what we're looking at here. All right. In this terminal end of the axon, this axon bulb, if you will, also sometimes called the axon terminal, there are vesicles with product inside them. And I'm just going to draw it like this. Okay. This is called a synaptic vesicle with containing whatever neurotransmitter inside. Neurotransmitter is a chemical. That's just generic, neurotransmitter. It can be a lot of different things. It could be dopamine, it could be serotonin, it could be norepinephrine, it can be acetylcholine, which is what it's going to be in this case. It depends on the synapse what the neurotransmitter is, but at a neuromuscular junction, the specific neurotransmitter is a chemical we call ACH, which stands for acetylcholine. All right. When the impulse comes down the axon, so the impulse is generated back here at the cell body, and we're going to get into that later when we get into the nervous system. The impulse is generated down here. Incidentally, the synaptic vesicles with the neurotransmitter are generated in the cell body too. They travel down the axon to live here. But the impulse is generated back here, and it travels down the axon like this. We call an impulse traveling down an axon an action potential. That's the name for it. We usually just abbreviate it AB. Okay. So the action potential is traveling down the axon, and when it gets to the axon bulb, that action potential actually causes some channels to open. And those channels specifically are for calcium. Now, calcium exists out here. It's in the extracellular environment with a lot of other ions, like sodium, for example. So when calcium channels in the axon bulb open, calcium naturally wants to move in because there's more of it outside the cell than inside the cell. So the action potential triggers an influx into the axon bulb of calcium ions. And that's important because these calcium ions stimulate these things to release their product. You have to have that step in order for them to release their product. So here's what happens. These vesicles, they move toward the very end of the axon bulb, and they kind of fuse with it. And through that process known as exocytosis, some of them are going to go like this. They're going to release this acetylcholine into this space. This space that the acetylcholine is released into is called a synaptic or the synaptic cleft. It's just the space between the axon and the skeletal muscle cell. We're going to zoom in on that space now, okay? We'll go over here, we'll zoom in. Oh. So we're at the very end of the axon bulb. And we're going to just draw, really expand the space, and just, here's the skeletal muscles. And so we know that the neurotransmitters released into this space, we have all these molecules of acetylcholine now in this space. All right, well, here's what happens next. There are receptors in the sarcolemma, right? It's called, that outer membrane of the muscle cell. Okay, that's that. There are receptors all in this membrane at the point of contact with the axon bulb that are specific for acetylcholine. And acetylcholine usually bonds in a couple places to these receptors, just like that. Now, they call sort of the region of the sarcolemma where these receptors are and where the acetylcholine bonds, they call that the motor end plate. Motor end plate. That's region of sarcolemma contacted by motor neuron axon, right? So if you, by definition, we said the synapse is the connection between two communicating cells. It's technically involves sort of three parts. It involves the axon bulb, the synaptic cleft, and the motor end plate. All of that is the synapse. Those are the components of the synapse. Okay, so what happens next is binding of acetylcholine to these receptors causes, and there's different mechanisms by which this happens, but this is the most common one. The receptor becomes a channel once that acetylcholine bonds to it. It's like acetylcholine bonds and then it opens up. The receptor literally opens and becomes a channel, okay? All right, so once that binding happens and the receptor opens up, the channel is very specific for a specific ion. And it's not calcium, and it's not acetylcholine, it's sodium, all right? Sodium, much like calcium, exists outside in the extracellular environment. So when channels open that are sodium channels, they allow sodium to traverse them. Sodium is going to come in from out here through these channels. You get the idea. So we get this influx of sodium into the skeletal muscle cell. That is going to cause something to happen to the skeletal muscle cell called depolarization. Skeletal muscle cell depolarizes as sodium enters. And what depolarizes means is the skeletal muscle cell becomes more positive. Which, if you look at this, that makes sense, right? We're allowing a bunch of positively charged ions to come into the muscle cell, right? The more sodium that comes in, the more positively charged this cell becomes. Would you agree with that? Does that make sense? Okay. So, it's not enough to just have some sodium come in. We actually have to have enough come in to reach a particular number, a magic number, if you will. That magic number of positivity is called the threshold, okay? And I think that was asked in the narrative or something you did, your worksheet. And I don't want to get too bogged down with it, really get too more complex than that, because we're going to revisit this and talk about it in more detail when we get to the nervous system. But just trust me, this is an important step that has to happen. You have to have enough sodium come in that the skeletal muscle cell reaches this certain peak of positivity, and when that number is reached, when enough sodium comes in, this signal that was coming down this axon literally is going to be regenerated right into this cell, specifically at the motor end plate. Not right away. We have to have enough sodium come in, but once we hit that threshold number, literally that action potential is recreated. All right, that's as far as we're going to go on that. So once the action potential is recreated, we need that signal to get down into the depths of our muscle cell to start making things happen. So hopefully, I haven't seen you in a couple weeks, but you've been reading a lot about the design of muscle cells. There was a video on it. Obviously, it's in the presentation. I asked y'all to be pretty versed on muscle cell anatomy and design when you got here. On the inside of the sarcolemma, coming literally off of it and going deep down into the cell, is a series of tubes called the transverse tubule network. These tubes originate from the inside inner part of the sarcolemma. So remember, the sarcolemma is the membrane around the cell. On the inside of that membrane, these tubes come off, and they literally carry the impulse that started here and was regenerated here. They carry the impulse down into the depths of the cell, okay? So once the impulse is moving down the transverse tubules, the whole point is to get the impulse to the sarcoplasmic reticulum. That's where we want the impulse to get to. The sarcoplasmic reticulum, which is just the endoplasmic reticulum with a fancy name, is surrounding those myofibrils that we talked about 2 weeks ago that are found inside muscle cells. So, quick recap, you've got these long muscle cells, right? Long cylindrical muscle cells, and they have organelles in them, nucleus, mitochondria, all the normal stuff. But they have these additional long, thin cylindrical organelle-like structures called myofibrils, remember? And like if I pull one out, it looks kind of like this. This is a myofibril. Okay. So myofibrils mimic the shape and the length of the muscle cell, but they're just much skinnier. And so we had an analogy when a couple weeks ago where I said, you know, if you had like one of those old-timey straw containers, you know, that are cylindrical and tall, they sit on a diner, you know, countertop. The container itself would represent a muscle cell, and then all the straws inside would be like these myofibrils. Okay. So contraction happens at the level of these, and so that's why we want to understand what they are. Back to sarcoplasmic reticulum. The location of the sarcoplasmic reticulum is closely associated with these. It's as if, and again, I like to make analogies in my brain, it helps me to understand too. And most of us are women, so we'll get this, and we'll explain it to the gentleman here. You got it. Fishnet stockings, right? You've all seen them. Maybe worn them with a Halloween costume. Mm-hmm. Maybe you're a can-can dancer. I don't know. But fishnet stockings. Imagine your leg is like a myofibril, and you're putting your fishnet stockings on, and it's like this netting all over your leg is what it looks like. That's how the sarcoplasmic reticulum exists around this myofibril. It's just completely surrounding it, like a pair of stockings, okay? The T tubules carry the message to the sarcoplasmic reticulum that's associated with every myofibril. And once the message gets to the sarcoplasmic reticulum, it responds to the action potential by releasing calcium. We're back to calcium again, okay? Calcium is stored in the sarcoplasmic reticulum of skeletal muscle, and it will be released from the SR in response to the action potential. All right, everybody with me so far? Okay, now we want to look at why do we care? What does that calcium do? Okay, so let me erase this. If you want to take a picture, do so now. I'm going to erase it and start doing some other stuff. This is all in the presentation. Please, please, please. I think after I finish talking today, that sliding filament presentation is going to make a lot more sense when you go to look at it, and then the videos as well. Okay, so I want to remind you of the design of the myofibril. I'm going to leave that. The myofibrils that make up each of the cells are the structures that are concentrated with protein. And they are composed of the filaments, the myofilaments, that are going to slide past one another when we think of the concept of the sliding filament theory. Okay, so let's go over here. I'm going to draw it kind of big. This is a myofibril. And you know, it goes on long. Okay. The myofibril, if you recall, is made up of repeating segments, okay? And each repeating segment, the boundary of each one is called a Z-line or Z-disc. And this repeats in a predictable way all the way along the length of the myofibril. The protein arrangement is such that, and remember, what I'm drawing right now is like one of these long myofibrils with these sections, okay? So these are cylindrical. I can only draw it in 2 dimensions, but imagine it's coming out of the board and going into the board. Attached to each Z disc is a filament known as actin, or the thin filament. You want to be as symmetrical as you can when you're practicing kind of drawing this and understanding this. That is called actin, also known as the thin filament, because it's thinner than the other one. And then over here, they usually kind of go in between these two to draw the actin here. Like that, and then this one would be this. And we keep going. In between the thin filaments, drawing it like this, is another protein again, the symmetry is important. So it would be like that. And up here we might draw it like we're seeing a little bit of this one. Notice how I'm making sure that this filament I'm drawing here does have overlap with the actin on the sides. I'm not drawing it like this. That's incorrect. This filament has to be long enough that it overlaps with the actin on each side. And so forth. All right, this is called myosin, also known as the thicker or thick filament. Together, these are referred to as the myofilaments of one Z to another Z. Remember what that's called? Sarcomere. That's called a sarcomere. Okay? And we're going to look at contraction at the level of a sarcomere. At the level of one of these toilet rolls. But there are hundreds along the length of the myofibril. But we're going to look at just one and imagine that if it's doing what I show you it's going to do and all of them are doing that, then the muscle is shortening. Okay. Okay, so I want to show you, when I draw it like this, it's like a schematic, I draw it kind of boxy and everything. I want to give you a little more insight into what actin and myosin actually look like. So, actin is a double-stranded protein. That's actin. And it has two other proteins associated with it. They are not part of actin, they are just associated with the actin. All right, one of them, it's like kind of woven like in between. I just sort of draw it like that. That's called tropomyosin. So imagine you took some yarn and you just twisted two different colors together, okay? The actin is in orange, tropomyosin I've drawn in purple. There's a third protein that is sort of holding the tropomyosin in position, and it's called troponin. Now sometimes they'll draw it like that, almost like a little snowman. It's a 3 subunit protein, it's got 3 parts to it. Sometimes you'll see it drawn like a little pyramid. It doesn't matter. How do you know? But it's, you know, scattered all along the length of the actin filament. And again, it's holding the tropomyosin in place. I think of it as like glue. It's like glue holding it in place. Okay, we're going to come back to that. Here's what myosin looks like. Myosin, this region of myosin that you see in between the actin looks like that. The myosin is hundreds of myosin molecules all clustered together. That's why it's so thick. It's not just like one strand of myosin. It's hundreds, like 400 something, I think. Here's what myosin looks like. One molecule looks like this. Myosin has a double-stranded tail, and then at one end, it's got these 2 heads like that. That's basically what a molecule of myosin looks like. If I want to translate this to here, here's what it looks like. It's like you have tons of these with these heads. And then maybe here's another one and the heads are going down like that. Here's one and the heads are going out like that. Here's another one, the heads are like this. And these heads are also coming out of the board and into the board. It's like a curling brush, right? Where you've got the handle, you know, that's this part. And then all of the little, what are those called? Bristles. That's like the myosin heads. And it's like if I took 2 of those brushes and put the ends together and then you have the bristles out here. Sort of think of it like that. Does that make sense? Okay, so then you're gonna have some other molecules oriented the other way that are going like this. And with their heads like this. You get the idea. Like that. Okay. So that's what this blue bar represents. Hundreds of myosin molecules with heads reaching all around. Okay. And then the actin is thinner and it looks something like that. Cool? Okay. Here's what happens. So we said that when the impulse reaches the sarcoplasmic reticulum releases calcium. Right? Okay. That's important because here's what the calcium does. The calcium goes and bonds to troponin. when it's released from the SR. Now remember, troponin was acting like glue. It was holding tropomyosin in place, in a particular place. But because tropomyosin needs to be in a particular place because there are these binding sites that it's basically covering up along the actin double strand that are available to bond with myosin. But when you're not contracting a muscle, I don't want the two bonding, or you're gonna get contraction that is uncomfortable and unwanted, right? So when you're relaxed, tropomyosin is in position to prevent myosin and actin from bonding. But here's what happens. When calcium bonds to troponin, you've got this double strand. I know I could do better, but whatever. It's like tropomyosin shifts out of the way. Troponin lets it go. And now these binding sites on actin are available for these heads of myosin to grab on to them. You see what I mean? So we release calcium from sarcoplasmic reticulum, it binds to troponin, that causes tropomyosin to shift, and now actin is available to bond with myosin. Let's talk about myosin for a minute. Myosin, these heads specifically, obviously are going to be involved in binding or bonding with actin, but they are also enzymatic. These heads perform an enzymatic function. The heads function not only to bond, but also as what we call an ATPase. And that ASE on the end, that tells you it's an enzyme, right? You know that from bio? Yes. Okay, so an ATPase is going to split ATP into ADP plus inorganic phosphate. And what do we get when we break ATP apart? What happens when we break ATP apart? What do we generate? Energy. So this is happening regardless of what's going on with calcium and troponin and all that. Your myosin heads are always attracting ATP and breaking it down to create energy. So the way I think of it is like those myosin heads are sitting there almost like shaking, like full of energy. They're ready. And they're just looking around, looking up, looking down, looking out, looking behind. They're like, where's the dang actin? When can I bind to it? They're sitting here with all this potential energy. So the minute that tropomyosin shifts out of the way and actin becomes available, those myosin heads are like, whoop, they just grab. They grab on to the actin filament. And they form what's called a cross bridge. So some of the heads reach up, some of them reach down, some of them reach forward, some of them reach back. They reach all around the cylindrical sarcomere. And they grab onto actin, and then their head shifts position. We form that cross bridge, and then the head goes like this. That's called a power stroke. And it pulls the actin and the Z line this way. And then the heads are going to let go. A new ATP comes in, they split it apart for energy, they recock their head, and then they grab again, and they power stroke, and then they let go, and then they reset. New ATP, energy, cross bridge, power stroke, release, reset. Cross bridge, power stroke, release, reset. And every time that happens, your muscle is getting shorter and shorter and shorter. That's contraction. And that's going to happen as long as that motor neuron is firing, and acetylcholine is in the cleft, and sodium is rushing into the muscle cell, depolarizing it to threshold, and all those steps we talked about are happening. SR is releasing calcium, it's bonding to troponin. As long as actin is available, Myosin is just gonna keep grabbing it and pulling it and pulling it and pulling it, like that. And the muscle sarcomeres are gonna get smaller. That means the myofibrils are getting shorter and smaller. That means the muscle cell is getting shorter, smaller. If we add all the muscle cells together, your whole muscle is getting smaller and shorter. Once we're done, and the motor neuron stops firing, and the acetylcholine, some of which is sort of sucked back into the axon, some dissipates out into the ether, into your extracellular space, some is broken down by an enzyme called acetylcholinesterase, but we get rid of that, everything just stops. Calcium is reabsorbed in the sarcoplasmic reticulum. It's no longer available to bind troponin. Troponin grabs back on to tropomyosin. We recover the active sites, and contraction stops. And those shortened sarcomeres just nice and gradually go back to their starting position once the sequence is not being initiated anymore. Questions about this part? Does it make sense? I know it's a lot of steps, but did the steps make sense? Did I lose you anywhere? Okay, let me show you what this looks like. Let me show you. I'm going to draw a big sarcomere that is relaxed. All right, so, we've got, I'm just going to do a very linear type of diagram. If I draw my Z lines like this, and I tell you, that a sarcomere is from Z to Z. Don't ever label a sarcomere like out here. You want to always label it on this horizontal axis so that it is clear you understand that it goes from Z line to Z line. Don't label it out here. That doesn't make sense. Okay. All right, so when we are drawing a sarcomere, which I've said before, you want to be really symmetrical. You don't want to draw like one actin like that. That's not true. It needs to be symmetrical. You do the best. That's pretty good. Okay. And we know that the myosin is the thicker one. It has to overlap with the actin on each side. Like that. And the myosin is very symmetrical. I'm not going to draw, a myosin like down here that's only that long. It's got to match the one above. Okay. Okay, so this is going to represent our relaxed sarcomere. Nothing's happening. Okay. But now I want to show you what happens when we contract this sarcomere. But before I do that, I'm going to label a few things on this schematic drawing, okay? And remember, there's another sarcomere continuing this way, that way, and then there's another one going this way, okay? But we're just looking at this one. All right, this area on the lateral sides of each sarcomere where there's only actin. If you look down here, there's basically, if we're looking, and I'm talking about comparing like actin and myosin, this area, if I did like a dotted line, like that, this is called the I band. It is defined as the region of the sarcomere where there's only actin. Now, sometimes they'll draw the I band kind of like this, and they'll say it's this whole thing because it's including the I band of this sarcomere and the I band of this one because they're right next to each other. But if you're just looking at one sarcomere, it's this lateral edges of the sarcomere. That's called the I band. I band. Region of only actin. All right, in between the two I bands, that thicker area, again, just think of it as in between the two I bands. This is called the A band. The A band is defined as the region of overlap between actin and myosin. Now, I understand there's no overlap here in the center, but since there's overlap between the two filaments here and way over here, that whole central region is called the A band, and that's its definition, the region of overlap between actin and myosin. But obviously at the center of the A band, would you agree there's a region where there's only myosin, right? So at the center of the A, if I get a little dotted line like this, it said just here where there's only myosin, this is called the H zone. The H zone is the region of only myosin. Okay. I'm going to draw in two other things that are not really bands, but just two other like labeling things we can identify. At the very center of the myosin, center of the sarcomere, rather, running through the very center of the myosin, if you looked at it under the microscope, is a very dark line of thickened myosin protein. It's all along the center of the sarcomere. This is called the M line. And again, it's a thickened area of myosin that runs down like through the entire center of the sarcomere. And it sort of holds the myosin in place. It assists in holding it in place because the actin is attached directly to the Z line, right? So it's held in place by its direct connection to the Z line. But the way we've drawn this several times, it's like the myosin's just floating there, right? Well, we don't want it to like move to the right or move to the left or go up or down. Like we want it to stay where it is, but it doesn't have like a direct connection. So at the center of the sarcomere, the myosin thickens and it anchors it through the center of the sarcomere, the cylindrical sarcomere, kind of holds it in place. There's an additional protein that is not a myofilament, not considered a myofilament like actin and myosin. There's an additional protein that helps assist in holding the myosin in place. It extends laterally from the edge of the myosin to the Z line like this. It's elastic in nature because it's going to shorten and it allows it to go back to its normal resting state. This is called titin, and it's Titin, and it's another anchoring protein. FYI, it's not a defining protein of the I band. When you're asked what is contained in the I band, it's just actin, okay? Because the bands reference the myofilaments. They're not talking about anything else. Okay, the banding pattern is relative to actin and myosin, not titin or anything else. But titin does exist, and it's just a protein that holds the edges of the myosin to the Z line so that it doesn't move around too much. Okay, so the reason I wanted to point out this banding pattern is because it's going to change with contraction. And you want to, kind of appreciating that, being able to replicate that, recognize that, is a way for me to know you really understand contraction and what's going on. So I'm going to erase this stuff down here. I'll just put that in the presentation. And I'm going to draw underneath my diagram up there, I'm going to draw a contracted sarcomere. All right. If I want to show what this sarcomere looks like when it's shortened, when it's contracted, the very first thing I want to do is draw Z lines and show the change that's happened in the position of the Z lines. So the Z lines started here and here. When this sarcomere contracts, they're going to move in, right? So that's always your starting point. I'm just going to draw them here and just show that they've moved inward. Okay, so now my Z lines are here, and then I can even go ahead and label my sarcomere And I can see right off the bat that my sarcomere went from being this long to being this long. I've done a lot of the answer right there. I've gotten some points because I've showed that during contraction the sarcomere shortens. All right, the next thing I'm going to draw in is the myosin. Myosin is not moving at all. It's moving things around it like a little puppet master, but itself is not moving. It's sitting there grabbing onto things and pulling things inward, pulling things inward, pulling things inward, but it's stationary. So myosin looks the same here as it does here. There's no difference. It doesn't get shorter or longer, doesn't change position. When you are drawing another contracted sarcomere, the myosin should be exactly the same as it is here, and that's why you always want to draw them vertically like this so you can compare them to each other. I wouldn't draw a contracted sarcomere out here because I can't compare that easily to this one. I draw it underneath. You see what I mean? Okay, so I'm just going to be as purposeful as possible and draw my myosin in here, keeping it the same length as it was above, you know, relatively. It's the same. Okay. My actin isn't shortening or anything either. It's just being moved in. It's called the sliding filament theory. That's what's happening. The filaments are sliding past one another. The myosin is causing the sliding because it's grabbing actin and pulling it in. But the actin is the same length that it is here. It's not getting longer or shorter. It's just moving inward. So, however long I drew the actin here, I'm going to draw it that same length here, give or take. I'm doing the best I can. But what I'm not going to do is draw the actin like that, right? That would make no sense. It's the same length as it was. It's just the Z lines have moved in, and the actin has been drawn closer to the center of the sarcomere because the myosin has grabbed it and pulled it inward, right? So it looks like that. All right, so now I want to look at the bands and see what's happened. So the first one let's look at is the I band, which we said is the lateral edge where there's just actin. And now it got a lot smaller, didn't it? It went from being this big to this big. So I smaller. Well, we already said the sarcomere definitely is shorter. Okay. Let's look at the A band. So we said up here the A band is the region between the two I bands. It's also another way of thinking about the A band is it's always the width of the myosin. Whatever the myosin is, that's what the A band is. So the A band goes from the edge of myosin to the edge of myosin. Well, did myosin change? We said it didn't get shorter or anything like that. So A should look the exact same. It doesn't change. We have more areas of overlap now, but the region of overlap didn't get smaller. So A stays same. All right, and then the H zone was the area of only, you could draw in the M line. But the H zone is the center of the A where there's only myosin. Well, it got a lot smaller, right? And incidentally, with maximum contraction of certain muscles, the H zone can go away completely. You can actually slide the filaments so far toward each other, like say my hands are active, that they overlap a little bit, and the H zone can disappear. It can with maximum contraction. But it definitely gets smaller. So these are sort of the things you want to be able to recognize, recreate, and understand. I is smaller, sarcomere is shorter. A band stays the same, and the H zone gets smaller. Contracted, relaxed. Question? Cool? Yeah? Okay, so go through that presentation now in the Unit 2 module that just says sliding filament, and you can click through that like a presentation. Like when you open the PowerPoint, go up to slideshow or whatever and play it, and it brings the steps up kind of one by one, where you can take it slow, read what's going on, look at the picture, make it make sense, and then go to the next step. Okay, it does a pretty good job, I think. Okay, Yulissa, will you stop the video?